In a remarkable demonstration of advancement in space exploration, NASA's private moon fleet is poised to accomplish 2 lunar landings within a single week, marking a significant milestone in the ongoing quest to explore Earth's celestial neighbor. Scheduled for Sunday and Thursday, these missions come at a time when interest in lunar exploration continues to surge, driven by technological innovations and the growing involvement of private enterprises in space travel. The upcoming landings underscore the collaboration between NASA and private companies, an approach that has become increasingly prevalent in recent years. This synergy allows for the leveraging of diverse capabilities, accelerating the pace of space missions while optimizing costs. As noted by a NASA spokesperson, this partnership not only enhances our exploratory efforts but also spurs technological advancements that benefit a wide range of applications on Earth. In addition to these lunar endeavors, there are significant developments in other sectors of science and health. The Centers for Disease Control and Prevention (CDC) recently reported a concerning rise in measles cases, highlighting the infectiousness of this virus, which can spread through simple respiratory activities such as sneezing or coughing. The outbreak has claimed its first U.S. measles death in over a decade, emphasizing the critical need for public health initiatives aimed at vaccination and awareness. Measles remains one of the most contagious diseases known, and it is vital for communities to ensure high vaccination rates to prevent outbreaks, emphasized a CDC official. The rise in measles cases is part of a broader public health challenge as health officials grapple with various infectious diseases, including a particularly harsh flu season that has led to high mortality rates among children. The CDC estimates that approximately 19,000 individuals have succumbed to the flu this winter, raising alarms about the potential long-term effects on public health systems. Meanwhile, the World Health Organization (WHO) is investigating over 1,000 illnesses in a village in the Democratic Republic of the Congo where water contamination is suspected as a contributing factor. Health officials are working diligently to determine the cause and mitigate the spread of illness in this region, which has historically faced challenges related to health infrastructure and disease management. In the realm of education and workforce development, Ohio lawmakers are introducing new legislation aimed at requiring high school students to complete a computer science course prior to graduation. This initiative addresses a significant gap in education, as 38% of Ohio high schools currently lack any computer science offerings. As stated by Representative Gail Manning, one of the bill's sponsors, the goal is to equip students with vital skills essential for success in an increasingly technology-driven job market. Our economy relies on a workforce that is not only familiar with technology but also adept at using it creatively, she noted. The importance of such educational initiatives is underscored by the need for a tech-savvy workforce, particularly as artificial intelligence and other advanced technologies permeate various sectors. Chris Berry, president of Ohio X, remarked, "As AI becomes more ingrained in our businesses, we need workers who can innovate and lead in this new landscape." In heritage science, researchers are actively developing practices that adhere to fair, findable, accessible, interoperable, reusable data principles. This movement aligns with the broader Open Science Initiative, which promotes transparency and accessibility in research. A recent project conducted by the Heritage Laboratory at the Swedish National Heritage Board emphasized the necessity for tailored vocabularies and metadata practices that reflect the interdisciplinary nature of heritage studies. We must ensure that our data management practices are as robust as the scientific methods we employ, stated Asa M. Larson, one of the researchers involved. As the landscape of science and technology continues to evolve, these developments showcase the interconnected nature of various fields, from space exploration to public health and education. The ongoing commitment to innovation, collaboration, and data accessibility is likely to yield significant benefits in the years to come, paving the way for enhanced research, better health outcomes, and a more informed public. Looking forward, The emphasis on technological literacy and open data practices will be crucial as society navigates the complexities of the modern world. In a significant development within the biopharmaceutical sector, Thermo Fisher Scientific has unveiled a new method for characterizing lipid components and lipid nanoparticles. LNPs, a crucial advancement for optimizing drug delivery systems, particularly in mRNA therapies. As the medical community continues to explore the potential of mRNA, from vaccines to personalized cancer treatments, the need for precise and reliable LNP formulations has never been more pressing. Lipid nanoparticles serve as protective carriers for mRNA, ensuring its stability during delivery to targeted cells. However, the intricate nature of LNPs poses challenges in their formulation, necessitating a careful balance of factors such as particle size, charge, and encapsulation efficiency. According to Dr. Ken Cook and Sis I. White from Thermo Fisher, the accurate characterization of LNP components is critical to ensuring that these formulations safely and effectively deliver therapeutic agents. One of the key technological advancements introduced by Thermo Fisher is the ultra-high-performance liquid chromatography-charged aerosol detector UHPLC-CAD. This sophisticated method allows for the rapid and precise determination of lipid molar ratios, enhancing both sensitivity and reproducibility under compliance-ready conditions. The innovation simplifies sample preparation and method development, promoting efficient quality control and expediting the pathway towards scalable production of LNP-based therapies. The complexities of LNP formulation were highlighted in a recent study that utilized advanced chromatographic techniques to quantify lipid components and their encapsulated payloads. The researchers specifically focused on polyadenilic acid potassium salt poly as the payload, employing the Thermo Scientific Ignite nanoassembler for formulation. The encapsulation efficiency was measured using the ribogreen assay, while the size of the LNPs was assessed through dynamic light scattering (DLS). Notably, initial attempts to separate lipid components faced setbacks due to the high concentration of solvents used, which compromised the integrity of the LNPs and resulted in poor data. However, the development of a refined method improved lipid separation and clarity, allowing for distinct detection of poly encapsulated in the nanoparticles. This breakthrough is not just a technical achievement, it represents a step forward in ensuring the reliability and precision of lipid quantification across various formulations. The implications of these findings extend far beyond academic interest. The UHPLC-CAD method has proven effective for measuring individual components of LNP formulations, with results showing strong agreement with original sample ratios. This reliability is paramount, as the characterization of drug products is essential for ensuring both safety and efficacy. The implications are profound, especially given the rise of mRNA technology in treating a range of conditions from infectious diseases to cancers. The success of this methodology could pave the way for more robust LNP formulations in clinical applications. With current FDA-approved mRNA vaccines for COVID-19, including mRNA 1273 and BNT162b serving as prime examples of LNP technology, the potential for future innovations remains vast. As researchers continue to push the boundaries of mRNA therapies, the enhanced capabilities for LNP characterization will likely play a critical role in the development of next-generation vaccines and treatments. In conclusion, as the biopharmaceutical landscape evolves, Thermo Fisher's advancements in LNP characterization signify a promising frontier in drug delivery systems. The ability to navigate the complexities of lipid formulations will be essential in harnessing the full potential of mRNA technology, ultimately leading to life-altering therapies and improved patient outcomes in the years to come. Recent developments in cancer research highlight both the challenges and advancements within the field. One notable study published in Scientific Reports sheds light on the intricate dynamics of cancer immunoscape, particularly the modulation of the tumor microenvironment. The research conducted by Pujan Shrestha, Zara S. Qureshi and Jason T. George introduces a generalized stochastic model that enhances our understanding of immunoediting in tumor immune coevolution, crucial for addressing metastatic disease, which remains the leading cause of cancer-related deaths. Despite significant advancements in therapeutic interventions, metastatic disease continues to pose substantial challenges in cancer treatment. The authors have noted that while previous models have focused on the adaptive immune system's role in combating tumors, there is a notable gap in understanding immune-mediated dormancy. This phenomenon allows the adaptive immune system to maintain a micrometastatic population, effectively delaying the emergence of clinically detectable metastasis. The research aims to quantify the effects of immunomodulation on T-cell-mediated cancer killing, employing a nonlinear birth-death model that accounts for various cytotoxic T-cell immunosuppressive effects. The model's findings indicate that disruptions in the balance between T-cell activity and tumor growth could lead to significant clinical implications. By examining bladder and breast cancer patients, the researchers developed a method to estimate immunomodulation's nature and extent, thus providing insights into disease recurrence timing. The implications of this work are profound, suggesting that a deeper understanding of tumor-immune interaction dynamics could lead to more effective identification and treatment strategies during the micrometastatic period. In a separate yet related incident, a cautionary tale emerged from a courtroom as a former cancer researcher at Stanford University was convicted for tampering with breast cancer research data. Nahid Manji, who had been dismissed from her position as a research coordinator, infamously replaced legitimate data with personal insults directed at her supervisor. This act of sabotage resulted in significant financial losses for the university and raised serious ethical concerns within the scientific community regarding data integrity and the repercussions of personal grievances that can lead to such destructive behavior. On a positive note, advancements in artificial intelligence are also making waves in cancer diagnostics. Researchers at the University of Jyväskylä have developed a new AI-based tool that significantly improves the classification of colorectal cancer tissue samples. This tool, which boasts an impressive accuracy rate of 96.74%, automates the analysis of tissue slides, potentially easing the workload of histopathologists and expediting diagnosis and treatment decisions. The availability of this AI tool aims to foster collaboration in cancer research, though its integration into clinical settings will require careful validation to meet regulatory standards. Moreover, a recent clinical trial conducted by Harvard researchers aimed to assess the impact of daily aspirin intake on breast cancer recurrence. Unfortunately, the trial concluded with no observable benefits, reaffirming the need for evidence-based approaches in determining effective treatments. The study emphasized that while lifestyle factors such as exercise, weight management, and diet play significant roles in cancer survival, adherence to prescribed treatment plans remains paramount. As these stories unfold, the landscape of cancer research continues to evolve, highlighting the importance of both innovative approaches and ethical considerations in advancing our understanding and treatment of this complex disease. Looking ahead, the integration of these insights into clinical practice could pave the way for improved patient outcomes, illustrating the ongoing commitment to combating cancer through scientific inquiry and ethical integrity. In a groundbreaking achievement that could reshape the future of biology and medicine, the 2024 Nobel Prize in Chemistry has been awarded to David Baker, Demis Hassabis, and John Jumper for their transformative work in protein structure prediction using artificial intelligence. This recognition underscores the monumental strides that AI has made in scientific research, with the Nobel Committee highlighting the enormous potential of their discoveries. David Baker, a biochemist from the University of Washington, received accolades for his innovative approach in creating entirely new proteins, which he achieved through advanced computational methods. Meanwhile, Hassabis and Jumper, affiliated with Google DeepMind, were recognized for their development of an AI model capable of predicting the intricate three-dimensional structures of proteins from their amino acid sequences. This significant breakthrough has effectively cracked a decades-long challenge in the field of biochemistry, enabling predictions for nearly all of the 200 million known proteins. The potential of their discoveries is enormous, the Nobel Committee stated, reflecting on the vast implications this work could have in various fields from drug discovery to vaccine development. Proteins made up of combinations of approximately 20 amino acids are fundamental to numerous biological processes acting as enzyme structural components and regulators within living organisms. Understanding their structures opens doors to novel therapeutic approaches and enhances our grasp of biological mechanisms. The AI program developed by Hasabi's and Jumper known as the AlphaFold Protein Structure Database serves as a crucial tool for researchers globally. By allowing instant access to predicted protein structures, it accelerates advancements in fundamental biology and related disciplines, fundamentally changing how scientists approach their work. Anna Wedel, a member of the Royal Swedish Academy of Sciences, emphasized the transformative nature of this breakthrough, describing it as a standalone breakthrough solving a traditional holy grail in physical chemistry. This year's Nobel Prize in Chemistry aligns with broader trends in scientific recognition, particularly the growing influence of AI in research. The simultaneous awarding of the Nobel Prize in Physics to Geoffrey Hinton and John Hopfield for their contributions to artificial neural networks further highlights this shift. David Pendlebury from Clarivate remarked on the boldness of the Nobel Foundation's choices, stating that recognizing AI's transformative role in both physics and chemistry within such a short timeframe is unprecedented. On the geopolitical front, the implications of such advancements cannot be overlooked. As the United States and China both vie for supremacy in technology, including artificial intelligence, the landscape of scientific collaboration and competition is rapidly evolving. Chinese authorities are reportedly cautioning top AI experts against traveling to the U.S., fearing that sensitive information about China's technological advancements could be at risk. This move highlights the tense dynamics between the two nations as they navigate a global tech race, particularly in sectors as impactful as AI. In a related context, U.S. Secretary of Energy Chris Wright recently characterized the AI race with China as akin to the Manhattan Project. During a visit to Oak Ridge National Laboratory, Wright emphasized the critical nature of this competition, aligning with the sentiments expressed by leading figures in the tech industry, including Greg Brockman, co-founder of OpenAI. This historical analogy underscores the urgency with which the U.S. must approach advancements in AI to maintain its competitive edge. As these developments unfold, the world watches closely. The convergence of AI with scientific discovery promises not only revolutionary advancements in healthcare and technology but also raises significant questions about ethics, international collaboration, and competition. The future of AI and research is poised to redefine our understanding of biology and beyond, with the potential to address some of humanity's most pressing challenges. In a remarkable leap toward the future of sustainable energy, a small town in central Washington state is poised to become a pivotal player in the nuclear fusion race. Helion Energy, an American fusion research company, is planning to build a groundbreaking 50-megawatt nuclear fusion power plant in Malaga, a community of just 2,000 residents. If successful, this facility could harness the same fusion processes that power the sun to generate enough energy to supply approximately 50,000 homes by 2028. The potential site for the power plant is strategically located near the Rock Island Dam along the Chelan County side of the Columbia River. While the exact location has not been finalized, Helion is currently in discussions with local officials regarding land owned by the Chelan Public Utility District (PUD). This ambitious project aims to not only provide clean carbon-free energy but also significantly enhance the local economy, which has traditionally relied on agriculture and tourism. Helion's CEO David Kirtley emphasized the transformative potential of fusion energy. 50 megawatts is a big first step of commercial-scale fusion, he stated in a conversation with Reuters. He noted that the revenue generated from this initial plant would be reinvested into developing additional power plants, aiming to accelerate the deployment of fusion energy both domestically and internationally. While the prospect of commercial fusion energy has been tantalizing for decades, practical implementation has remained elusive. Helion's timeline is ambitious, aiming to commence construction this summer and begin operations by 2028. This timeline positions Helion to potentially outpace competitors like Commonwealth Fusion Systems CFS, which is working on a larger 400 megawatt facility in Virginia expected to be operational by the early 2030s. The implications of Helion's success extend beyond local energy needs. Chelan PUD has expressed hope that this fusion project will diversify energy sources in the region, which has primarily relied on hydropower. The demand for clean energy is not going away and it's only growing, stated Kirk Hudson of Chelan PUD, highlighting the need for a more resilient energy portfolio. Initially, energy produced by the fusion plant will support a purchase agreement with Microsoft. But plans are in place for broader access to local customers over time. As the United States races to establish itself as a leader in fusion energy, it faces stiff competition from global players like China, which has invested heavily in fusion research. According to Jean-Paul Allain, leader of the U.S. Energy Department's Office of Fusion Energy Sciences, China is currently the front-runner in fusion energy development with an estimated investment of $1.5 billion annually compared to about $800 million from the Biden administration. In the broader context of energy innovation, Helion's project in Malaga represents a critical juncture in the pursuit of sustainable energy solutions. With around 40 American companies and several government labs in the mix, the race to harness fusion energy is intensifying. As Helion moves forward with its plans, its success could not only change the energy landscape of central Washington but also solidify the United States' position in the global energy arena. The coming years will be pivotal in determining whether fusion energy can transition from a theoretical concept to a practical reality that fulfills the world's growing energy demands.